The Huntington's disease was an inherited disorder that was discovered in 1872. It was discovered by George Huntington, who wrote an article about chorea. Chorea is a Latin and Greek word that is known as chorus or groups of dancers. It became known as a dancing disorder in the Middle Ages. People who had chorea, their muscles jerked and twitched. The HTT plays an important role in the nervous system like the brain and other developments before birth. It is found in many tissues, especially the brain. The protein stem cell is a chemical signal, transporting materials, attaching to proteins and other structures, and protecting the cell from self-destruction.
The common allele is at the top, and the mutated allele is at the bottom. Where the line starts is where the repeating of the three nucleotides starts. Double-stranded DNA from the common allele goes through the transcription of DNA into RNA. The double-stranded DNA splits. When reaching the activation site, the RNA strand is made from the top of the DNA strand. The RNA is going through the exit port now. The new RNA strand that will go into the ribosome is formed. The expanded allele is also double-stranded. The expanded DNA strand goes through the transcription rate as well. As you can see, it begins to split again. As it approaches the activation site, the RNA strand is formed. It as well goes through the exit port. Comparing the two alleles, the top allele is the common allele and the bottom allele is the mutated allele. They enter through the ribosome 3x3, attaching to tRNAs as a codon. The tRNAs, as you can see, the amino acids is attaching to one another and exiting through the exit port. It moves on from glycine to alanine. As you can see, this is the final outcome of the amino acid strand. Then the mutated allele goes through the ribosome, 3x3 as well, attaching to the tRNA, forming the amino acids that attach.
Alleles come from sexual reproduction because genes are produced with a partner and equal amount of DNA is inherited from two parents. Each parent contributes 23 chromosomes. Two copies of every gene inherits traits. The alleles may produce homozygous or heterozygous genotypes based on the possible combinations of the alleles. Then the phenotype will be determined based on the combination of the alleles. Huntington's disease has a pattern of autosomal dominant because a parent has one copy of mutated gene and the other one is a normal gene. One copy of the gene is sufficient to cause Huntington's disease. Additionally, the size of CAG through nuclei repeats increasing size of the gene. If a person has two mutated genes, they are most likely to die. If a person has a mutated gene, the gene is most likely to cause the disease. The phenotype of Huntington's disease is anticipation. For adults, the CAG repeats 40 to 50 times. For juveniles, more than 60 CAG repeats. Also, for people who have 27 through 35 CAG repeats don't develop CAG, although there's risk their children will develop the disorder. Punnett square. If you look at the Punnett square on the left, the Punnett square shows a parent with Huntington's disease and a normal parent. There's a probability of 50% that uppercase H and lowercase h will be produced, and 50% that lowercase hh will be produced. The possibility of having Huntington's disease is 50%. Although, if you look at the Punnett square in the right, the Punnett square shows both parents have Huntington's disease. So there's a 25% possibility that a person will be unaffected by the Huntington's disease, and there will be a 75% possibility that the person will have Huntington's disease because each parent is mutated. Pedigree. The pedigree demonstrates that blue circle is a female with the Huntington's disease, and the green square demonstrates a normal male. As you see in the pedigree, it starts off with a female affected by the disease and a male unaffected by the disease. Then the parents make children. The pink square represents male with Huntington's disease, and the yellow circle represents normal female. As you see in the pedigree, the blue circle, which is the female with Huntington's disease, affected the pink square, which is a male, and another female with Huntington's disease. Then the pink square on the left makes 2 more children over time. Although the male that is affected with Huntington's disease make a normal female and another male affected with Huntington's disease, the side to the right has a normal female which makes 2 more children. The normal female makes another normal female and normal male. This shows how the normal female did not affect the children with the disease because she had no mutations. The picture of the parents. The male parent is affected with Huntington's disease and the female parent is unaffected. The male parent has a mutation in one of his genes. The female has no mutations. When they have children, the father will affect 2 children, a girl and a boy. The 2 children in color blue will have a mutation which will have Huntington's disease. Having a mutation in a gene enough to cause a person having Huntington's disease. The other 2 children in color pink will be unaffected because they have no mutation since the mother was unaffected as well.